the sites here today. I think this is really a one of a kind type of place, and all the students are really, and the faculty should be really feel lucky to be in this space and be part of this ecosystem. It really uh, sort of puts at play the connection between industry and academe, and we always talk about it, but this is really happening right here. So congratulations on a very, very nice setup here. So what I'd like to do today is really showcase how we deal with gene and cell therapy in the US, specifically how academic medical centers such as ours basically try to sort of integrate um, gene and cell therapy work within their treatment system. So, if we define gene and cell therapies, I think we take a very broad view about it. And in terms of gene therapy, genetic modification, whether using DNA or RNA, via vector or other types of delivery systems, and there's different ways of thinking about how you want to disease uh, change, decrease expression of disease causing variants of proteins. You want to increase disease fighting proteins or deficient ones, and then add new and modified proteins in certain instances. Cell therapy clearly is taken off with CAR T because CAR T is part of cell therapy. But again, the recognition that there's different ways of delivering autologous or allogeneic cells has kind of redefined the field in the way we think about it. If we look at the approvals of both gene cell therapies, I mean, it's continuing to increase over the years, and what we see is just an exponential curve going up since 2019, 2018, and this is supposed to increase over the next decade or so. The GCT landscape and clinical trial activity today is basically shown here. Um, we've got about 1,400 GCT in preclinical stages, mostly focused on oncology and rare diseases. In terms of cell therapy, uh, we have a number of phase 2 and phase 3 trials now, especially in the areas of Parkinson's, type 1 diabetes, and osteoarthritis. And RNA therapies have really taken off with the modified RNA being used in the vaccines, in the COVID vaccines, but we're seeing a lot of play, as I'll show you, with RNA therapies. They're focused on other disease states, such as Duchenne muscular dystrophy, amyotrophic lateral sclerosis, and Huntington disease. Now, the recent successes in GCT landscape really builds the momentum for us for more approvals and innovation in the next few years. Specifically, I think some of the important approvals that we've seen are for Duchenne muscular dystrophy with AV9 microdystrophin, uh, first time cell gene editing being used for sickle cell disease. And that's really been sort of a game changer in the way we see gene editing in the clinic. So I think this has been a really big uh, uh, change in the field and a big advancement and setting up in vivo gene editing. Now, CAR T continue to dominate the pipeline for uh, gene and cell therapy. About 50% of genetically modified cell therapies are basically CAR Ts. There's also CAR NK and other types of therapies that come down, but really this is the one that's defining the cell therapy field. Now, interestingly, oncology and targeting by CAR T is mainly the focus of CAR T therapy. However, we're seeing an expanding new areas in autoimmune diseases and infectious diseases. So I think in the next few years, you're going to see the breakdown, the red triangle becoming bigger and bigger. Now, CAR T sets a very high efficacy bar in hematology, especially in some of the malignancies, such as lymphoma, leukemias. And one of the areas that we see a deficiency in CAR T is in solid tumors. There's been sort of very uh, sorry. There's been quite a bit of work on solid tumors using the auto CAR Ts with CD19 and BCMA CAR Ts, but unfortunately, the effectiveness in solid tumors has been very disappointing. What I want to show is that we at the Mass General Hospital, we have one of our experts, Marcella Maus, who has been manipulating the CAR Ts to be also not only targeting CD19, but to be secreting different types of promoters and growth factors, such as FGF in this case, and has been able to effectively target one of the deadliest cancer, and that's glioblastoma. And delivery of CAR Ts within the glioblastoma has really resulted in reversal in at least a couple of the three patients who were injected. So I think we're seeing resurgence of more focused and more complex type of CAR Ts. They're going to be able to hopefully address some of the issues with the solid tumors. Now, if you look at gene therapy, in terms of the vectors, again, a big portion of gene therapies are focused on rare diseases. Cancer continues to be a big area or big target for gene therapy. Neurological diseases also have had a big imprint on gene therapy, and cardiovascular much less. But the rare disease part is really important because you can basically equate a genetic mutation with a therapeutic that can correct for that mutation. I want to show you one example for really some of the dramatic changes you see with gene therapy. This is by one of my colleagues, Chris Bankowitz, who is a neurosurgeon. And here he's addressing a deficiency in kids of AADC. It's an enzyme that converts levodopa to dopamine. And the kids basically have very much Parkinsonian type of symptoms in terms of hypotonia, hypokinesia, dystonia, and irritability with some disturbances in intellectual abilities. So he's injected this AAV2 AADC into the brain of these kids with this disorder, and this is the some of the results. So this is a patient who has this disorder, and this is a baseline. As you can see, she can't stand by herself. and is unable to walk, has to be sort of lifted. Uh, 20-month post gene therapy, uh, kid is able to stand up and walk, and many months later, she's able to ski. So you can really see this dramatic evolution with one single injection in the substantia nigra of these vectors in these patients. So I was going to skip this portion, but some of you have asked me to sort of showcase some of the cardiovascular gene therapy, which I did this morning. So I'll be very brief on the cardiovascular gene therapy, even though this is exactly my field of work. And I apologize for people who were there this morning. I'm just going to give just a few slides since many of you here asked me to sort of recap some of the work that we did in cardiac gene therapy. So my focus clinically is on heart failure and cardiac transplantation. So I basically see many of these patients for over years. And one thing about congestive heart failure is that the incidence of heart failure continues to increase over time. And this is due to the aging population and the fact that we treat heart attacks much better now. So we really do have an increase in the incidence of heart failure in the US, in the EU, and worldwide. One of the striking things about this disease is that if you compare it to um, many of the cancers in men and women, it's very similar to some of the deadliest cancers that we have in men, such as prostate cancer, lung cancer, colorectal cancer, and also it's in women, it's comparable to some of the deadliest cancers in women, such as ovarian cancer uh, and breast cancer. So it continues to be a big epidemic. So we've taken a very specific um, approach in gene therapy for heart failure, where we um, look at what happens to the heart after an injury, such as ischemia, such as a heart attack, for example, infection, inflammation, uh, non-treated hypertension, other causes that lead to this advanced heart failure, where you have dysfunctional cardiac cells and abnormalities in the function of these cells. And what we've seen over the years is that we've kind of dissected down the molecular pathways that undo the cardiac cells and make it abnormal. And this is what we call a pathway disease, or targeting by pathways. And abnormal calcium cycling internally with decreased circuit activity and increased protein phosphatase 1 contribute significantly to what we call a abnormal contractile function in these failing hearts. So because of that, we have abnormal phosphorylation of different proteins, and one of the key activators or key players in the calcium handling is this specific protein known as oops, circuit 2A, which is an important pump in the cardiac cell that regulates calcium. And what we do is basically we're able to um, manipulate protein phosphatase 1 so that it enhances circuit activity in the cardiac cell. And we couple that with a vector that's very strong for a cardiac application, and that's the adeno-associated. And we'll talk a little bit more about some of the RNA therapies a little bit later. But AV vectors for cardiovascular application has really been a game changer. It's a small 20 nanometer particle that contains no viral genes. It's a helper-dependent parvovirus. You can see how small it is. This is an adenovirus, which is about 100 nanometers, and these are the AAVs. That's about 20 nanometers. For the heart, it causes a broad distribution to the myocardium, and it has different serotypes. So it's a single-stranded DNA, as you can see here, that is unwrapped in the nucleus, and it undergoes double strand, and then message is produced here. So we have different types of AV serotypes, and naturally occurring AV serotypes can have different tropism. The transgene within it can be around up to about 4.9 to 5 kb. But one of the aspects that's important is the fact that you can choose the vector that you want or the serotype that you want to target the liver, the eye, muscles, and so on. You can also play around with the capsid to make it even more specific or more tropic. In this case, we made the capsid very specific to the heart by manipulating the amino acids here. And you can see here in the humans, you can get one viral genome per diploid cell, which means you've basically saturated the myocardium with the vector, so about 100% efficiency. And this is in human hearts. Now, the liver, especially since we're talking about we're in the animal studies department. I wanted to show you how we sort of developed models based on many large animal experiments. So you can give vectors by intravenous injection, and that's sort of the simplest way. But what we do, what you have to do in this case, systemic delivery requires a large number of vectors, and that really is not cost effective and causes a lot of side effects. So using manipulation with cardiac catheters, we can deliver locally through retrograde or direct injection, or just simply by antegrade injection. And this is what we do clinically, actually, in patients. We inject down the coronary arteries. These are the arteries that feed the heart. This is the left side, the left main, and this is the right coronary artery. And we basically inject through pumps shown here in this movie, where we cannulate the coronary artery and inject downstream. So we conducted the first in-human trials, one of the 4 or 5 trials that we have in cardiac gene therapy that came out from the lab. And this is one of them where we used 2 different doses of this 2I8 I1C, which blocks protein phosphatase 1. And we see here a nice distribution of improvement in different parameters. So for heart failure cardiologists, New York Heart Association, ejection fraction, other parameters, such as VO2 max, 6-minute walk test, Minnesota Living with Heart Failure questionnaire, all are important. So in this case, you can see that the low dose seems to do a little bit better than the high dose. So red means worsening function, whereas the green is improvement, and purple is staying the same. In these type of patients, you would literally see after 12 months full of red, because these patients at baseline have a very negative path. So we have a phase 2 trial that's ongoing internationally in patients with non-ischemic heart failure, where we give low dose and high dose for these patients. So coming back to the GCT, one of the aspects that we think we always deal with surrounds really this we call the valley of death, because academia is able to generate a lot of targets. We're able to sort of bring out multiple different targets because we understand the molecular biology of diseases. We understand, obviously, the genetic mutations that are associated with hereditary type of diseases. But how do you travel through this valley of death, which is uh, the issue that we have. And the challenges in GCT translation comes from costly manufacturing and scale up, stringent regulatory frameworks. I mean, the FDA, obviously, difficulty in getting funding. Sometimes you have low commercial viability if your hereditary disease is only affects 10 patients in the world. Complex pricing and reimbursement, and sometimes, like we're seeing for the sickle cell disease, which is a really curative approach, is limited patient access and acceptance. So, how do academic medical centers and universities help in translating gene and cell therapy from bench to bedside? And to really travel this translational canyon, uh, we really have to sort of think about how to bridge uh, our investigators from in vitro studies and animal studies all the way to phase 1 studies, because that's really where you can see the safety of the product that you have. And we've come up with a number of approaches to basically help our investigators go from the lab to the clinic with these very innovative growth platforms and gene therapy. And we've called this the RISE model, which is really a framework for academic medical centers to foster gene and cell therapy advancement. And this was just published last week in Nature Biotechnology of how we at the Gene and Cell Therapy Institute at Mass General basically focus on improving this canyon. And this really goes around resource sharing, industry collaboration, sustainable funding, and education. And these basically come in a form that this implementation at Mass General Brigham GCTI is based on the fact that we have over 2,000 researchers, and of those 500 focus on gene and cell therapy. We have a large number of physicians at Mass General, and about a third of them are physicians, but they also go back to the lab and conduct basic research. And our hospital system cares for nearly 2.5 million patients annually, and we do see a variety of patients from the ultra rare diseases in neurology to the most common types of diseases like heart failure, diabetes, hypertension. So basically, you have the ingredients within our ecosystem, just within Mass General Brigham, to be able to take advantage of the research that's ongoing. So how do you bridge them? And the bridge is really how do we transfer technology from bed to bedside? And we've created a Center for Manufacturing Excellence so that we can become the suppliers of GMP products and teach manufacturing techniques, and also become the hub for fostering the commercialization of these products externally. And the way we've been working on this is that since we do have scientists and we have an engine for discovery, there's in vitro and in vivo research that are ongoing. We basically help with IND enabling preclinical work and the IND submission. And the GCTI manufacturing is able to basically bridge that gap of the manufacturing problem. And we've basically been able to partner with industry to get the best cost-effective GMP manufacturing. We have collaboration with multiple manufacturing platforms. So unlike a biotech, the institute has to deal with multiple different vector systems, as I'll show you, in different types of platforms. So we can't just say, all right, we have one platform, we're going to focus on this. We have multiple platforms. So we're constantly going back and forth about how we have to process develop process in sync with what we created multiple cores that basically feed into this process. And I'll go through some of these cores. But based on that, we're able to sort of leverage what we have within our system, whether it's cell therapy, IPS facility, vector facility, and the AAV vector core to be able to get to quality control and quality management for sending to the way of the patients. We also focus a lot on regulatory support. So we have a team of experts who guide the process in terms of FDA content preparation, FDA interaction, while at the same time making sure that the patents are filed and our investigators are protected in terms of their final product. So we've created multiple core services within the Gene and Cell Therapy Institute, and one of our favorite ones is the new RNA therapeutic core. We have a gene editing core that basically provides editors and guide RNAs and basically look at throughput screening for in vivo gene editing. We have tissue engineering cores where we create human engineered tissues, and these three-dimensional tissues have disease models that you can test, and they're really platforms for gene and cell therapies. And based on the Modernization Act that we have in the US, we're able to use these tissues for our IND enabling studies. So we don't have to do animal studies for pharmacology, but we can present these data of ex vivo human tissues as a surrogate for animal testing. And we have focus on different GMP facilities. So one of the areas that we really are very interested in is circular RNA technology, and that's part of the resource sharing that we have. We've built a circular RNA manufacturing core at GCTI, and these services are for the MGB community. We formulate with LNPs that are appropriate for these circular RNA. So why circular RNA, and why is it so powerful? So when we describe or develop mRNAs for therapeutics, especially the modified RNA, there's a cap structure that initiates ribosome scanning. So circular RNA doesn't have a cap. And basically, by having the circular RNA engage with the ribosome, you basically have a much more prolonged expression pattern, as you basically see here, whereby you have the activity of luciferase, which is being produced with the circular RNA, be extremely powerful. If you make the circular RNA pure, you make it really immunoquiescent. And this is important because if here's delivering circular RNA versus modified RNA, and you can see the IL-6 response in A549 cells or interferon response in the similar cells, whereas in modified RNA you have significant increase in these immunoactivators. With circular RNA, if they're pure enough, you don't. In addition, when you look at what happens to circular RNA, is that you basically can get a very robust expression with circular RNA that's much longer than what you get with modified RNA. Now, in vivo, one of the nice things that you see is that you're able to deliver the circular RNA repeatedly over time, as you can see here. Whereas with modified RNA, you just basically get a blip and then it comes down. With circular RNA, you can maintain high levels and repeatedly give this over many months in this animal model. So circular RNA really ticks off multiple boxes. You basically can have very nice peak expression. The duration is not obviously as long as some of these viruses, but it's definitely valuable for uh, short-term expression for a few weeks to a month. You can multiplex it, you can deliver it multiple times, and there's no immunity, and the cost is quite low compared to any other vector systems. And as a vaccine platform, it's been quite effective in terms of boosting the expression pattern of a variety of spike proteins, including the COVID protein. And this is basically showing its potential as a vaccine. Now, we've also been using it for rare genetic diseases where you can basically uh, uh, engage very large 12 kb circular RNAs for a variety of genetic diseases where, such as dystrophin, which is very large, and for cystic fibrosis, also the genes are extremely large. And what we're seeing here is the dystrophin gene being expressed in with circular RNA. Over, it's stable over a month. So it's
Cold storage? Cold. Minus 80s?
no, it's you can do it 4 degrees or 25 degrees. It's quite stable. Oh, I see. Yeah. No, so you can keep it on the shelf, basically. So we've also been using it as a immuno-oncology platforms, and basically the idea there is to you can deliver these in vivo uh, to basically enhance the T cells, and you can see that with this type of circular RNA that's delivering CAR, you can basically reduce the tumor burden. We've also been very intrigued by point of care type of approach, where you can basically have the circular RNA cargo be seated on these mechanotransduction devices, as we call them. They're called portals. And what happens there is that you basically have the T cells go through and be infected by the circular RNA that's uh, on these platforms, and you can get very effective cytotoxic responses in vivo using these activated T cells. And the idea is to basically take unstimulated T cells, or not having to basically go through the whole process of waiting for days and days uh, to get the auto CAR Ts, but to basically have a system where you take the blood out, isolate the T cells, and reinfuse it back after passing through this, because the expression pattern is very fast. It takes a few hours to get maximal expression. And this is basically shown here in our GFP expression in PBMCs. So this is one example of a gene editing, sort of a core, the circular RNA core that we've sort of leaned into because we feel that this is a disruptive technology that is now available to all of our MGB investigators. We started using it in September. We opened up the circular RNA, and there's been now over 200 orders placed for circular RNA within MGB and also outside in multiple research agreements with companies. So regarding the sustainability of our institute, we also have mechanism to uh, have what we call um, concept creation to investigation stage all the way to three different strategies of commercialization, licensing, in-house development, and company creation. And these, especially the last two, have to have an absolute input from our clinical teams because they're the ones who are driving this process. And we have commercialization hub where we can basically uh, have the process development, the farm talks, and GMP manufacturing all incorporated in the sort of circle of the institute. And we power that by being able to deliver grants to early stage concepts that would have the legs to go all the way to clinical stage. So we have research grants at the preclinical stage or the IND enabling stage at different levels that we give out every year. And what we do is basically once these groups receive these grants, we follow them like biotech companies. So we have board meetings with them. We give them advice on what to do next. And these are all based on drug products. If there's no drug products, they don't get funded. I mean, because this is going to be the most important part of our investments. And at certain stages are go or no go decisions about further funding them as we go forward. So this is the idea of these grants is that you basically invest into these programs, you expand the portfolio within our medical system, generate novel IP. There could be licensing or partnership or company creation, and then we reap the financial return on the investments. To give you an idea of some of the applicants that we get, they really span all kinds of diseases, from oncology to neurology to rare diseases, and different platforms. So we have CAR T grants, engineered cells, a lot of AAV-based. Oncology and neurology are very well represented here. Gene editing, platform-based RNA therapy. So we can see that there's a variety of platforms that we use and variety of diseases. So I think the future of gene and cell therapies, tremendous amount of progress, but the current gene and cell therapies are really exorbitant pricing, and that's really making it out of reach and giving a lot of governments the second thoughts about with limited resources, how can you sort of even think about gene and cell therapies for patients? Now, the complex nature of the biological processes for gene and cell therapies require significant manpower, and high doses for vectors drive up the costs. So, in countries around the world, as they sort of think about the gene and cell therapy, local-based raw materials need to be produced because that's really what some of the biggest costs that come in the US are based on plasmid DNAs, enzymes, all the solutions. And at the end of the day, innovation in vectors will lead to much lower doses and lower costs of goods. And I think, can't emphasize more how innovation is really critical in the expansion of gene and cell therapy into the world. So with that, I'd like to thank you for your attention. This is my group at the Gene Cell Therapy Institute. and we have different expertise, and each one of them lead small or big groups of other investigators. Thank you. Happy to take any questions. Yeah.
Was a nice presentation. So recently I came across this vaccine from Russia, like that will be very personalized and based on mRNA itself. Is it a circular one? And also like it has been heavily quoted by the Indian media in the South Asian countries. I did not saw any reporting from the West. So can you help invite to come have came across? Thank you.
Thanks. So, yeah, these new vaccines are, very specific to the modification of the RNA nucleic acids. Um, whereas circular RNA are quite different. So I'm actually, not aware of the specific example that you're talking about in terms of a vaccine. Is that in India or?
It's Russia. They are planning to provide it by 2025 free, of course.
Yeah. So that's a vaccine for, yes, I've heard about the vaccine for tumors. So there's been a lot of work on mRNA vaccines and tumors. And the way it works is that you isolate specific markers from the tumors that you have, and you produce guides that represent these by mRNA and you can give a cocktail of mRNAs. But it's very patient specific. So one tumor is going to be different from another tumor, even within the same class. So I'm not sure what this specific vaccine, the Russian vaccine, is all about, but I definitely have heard about it. So I thought it was something different. I've definitely heard about the vaccine that's going to come out next week or something in Russia. So we're all waiting to see what it is.
Thank you.
So yeah, actually there will be more questions, but the idea is that we do panel and
Yeah.
some questions will be answered in the panel already.
Yeah.
And then in the panel also they can ask again more. So let's start the panel now. And during the panel discussion, some of your questions will be answered, and even in between the panel also you will disturb and answer. Okay. Yeah. So let's stop questions for now, and we'll set up the panel, and then reserve your questions for that. Yeah. Stop. Yeah. Absolutely. Okay, thank you. Thank you, Roger. I mean, please reserve your questions. Please don't leave. We still have a panel discussion. So I would like to invite first our moderator, Dr. Uday Saxena, onto the stage. So Dr. Uday Saxena is the founder of IFER Foundation for Translation Research. The foundation trains women scientists in translation research, leadership, and entrepreneurship. Most recently, he also co-founded a company called Utopia Therapeutics, an innovation-driven company focused on vaccine and stem cell platforms. He's a co-founder of other startup company. I mean, his profile is so great, and many actually we have to dream for such profile. So, big pharma guy, like a previous CSO at Dr. Reddy's, and also many he has led Lipitor Discovery, one of the largest selling drug on in pharma space. So happy to have you, right? Thank you. So he will be leading the panel, and then we have Dhananjay, Dr. Dhananjay is coming. Okay, maybe I'll introduce once he's back. Yeah, we have Dr. Suresh Pusla here, the founder of Akasta Health Private Limited. He's a leader, scientist, and motivator with more than 3 decades of international experience in fostering science, innovation, collaborative culture, by building and nurturing high-performing teams. His career spans scientific excellence in discovery and research, which resulted in high-impact publications, in high visibility international journals, including Nature, and he created exceptional quality in vivo facilities and created trained teams for R&D. So he's done a lot of work, I think. Yeah, thank you, sir. Thank you for joining here. Yeah. And we have Subharanjan Chakraborty here, the head of technical operations at Vijay Biosciences, a biotechnology company, who has a robust background in research, accumulating 8 years of experience in academic settings and 13 years in the biotechnology industry. His work focuses on leading technical operations and fostering collaborations with innovative leaders in the field. You might be knowing his through BIRAC. He has played a big role in BIRAC. Yeah. Thank you, sir. Welcome here. And we have Dr. Nuruddin Khan here as a professor in the Department of Animal Biology at the School of Life Sciences at the University of Hyderabad. He received his PhD from Molecular Immunology from Manipal University, Center for DNA Fingerprinting, and CDFB. And his MSc is in Biotechnology from Jamia Hamdard University. Um, he has, I think currently, so you work a lot with the autoimmune issues as well as in the vaccine area as well. And psoriasis, we even Redana sir and Nuruddin sir has published recently about psoriasis. A patent was also delivered. Yeah. But is Dhananjay here? Yeah, Dhananjay, come. The other panelist we have is Dhananjay Navandar. So please, Dhananjay, come onto the stage. He received his BS in biotechnology from the University of Nebraska and completed his PhD in cellular and molecular biology at the University of Wisconsin-Madison. His doctoral research led to the identification of 2 host and 1 viral protein as novel therapeutic targets for lytic induction therapy to treat Epstein Barr virus positive tumors. He conducted his PDF at Dana-Farber Cancer Institute, where he developed a targeted DNA methyltransferase tool to selectively silence the expression of specific genes of therapeutic relevance. So currently he is heading Vijay Bio along with Roger. So he will explain more about Vijay Bio as well. Because I think we also have to focus a little bit because what Roger has explained is the RISE program there, what you are trying to do for Indian context as well. I think you should explain that. Okay. Now the stage is open for panel discussion. I request Uday to lead it here.
Thank you so much for staying back. As you just heard, we have a fantastic panel headed by Roger here. So the way I've designed the questions is to try and understand where US is in terms of cell, gene, and vaccine technologies, and where is India, and what are the gaps in between? What is it that we need to do in India to be able to fill those gaps? So really, that's the whole purpose, is to try and understand where they are and where we are, and see if we can move forward there. Actually, Roger's presentation was fantastic in terms of laying the grounds of what you're doing towards understanding of regulatory, commercial, ideation. I thought that was beautiful. So I think that was fantastic. I had sent these questions in advance. I hope you had a chance to look at them. If not, this is a good time to start thinking about it. So the format is that we will ask 3 questions of each. The same question will be asked to each of the panelists. We'll finish 1 question, come back for second, and then do the third one. I'll try and wrap up in about 35, 40 minutes, so that'll leave about 20 minutes or so for Q&A from the audience. So first of all, can you hear me well, or am I shouting or not shouting? Okay. So I'd like to start with our guest of honor today, Roger. The first question is, and you explained some of this beautifully, is what is the current status of cell molecular and mainly cell and gene therapies in the United States in terms of regulatory? I mean, how quickly is it that the FDA is going to adopt some of these technologies? I heard you say they were mainly oncology and rare diseases, which is basically, caught between a rock and a hard place type of situation. But more common diseases, when is the FDA going to allow you to adopt these for more common diseases?
Yeah. No, thank you. Yeah. So I think over the last 5 years, we have seen within the FDA a real, literally a seismic shift in terms of what is acceptable. So to give you an example, for example, in heart failure and cardiomyopathies, it was really survival. So you had to power your studies for hundreds, thousands of patients, so you're able to see 10, 20% survival benefits. with gene therapy and with cell therapy, the FDA has basically understood that you cannot conduct studies that are in the hundreds and thousands of patients. And what they've been accepting, like they did for Duchenne muscular dystrophy, are what's called surrogate endpoints. And these surrogate endpoints, clinical endpoints, that can be measured more easily. You need fewer patients to make your point. Can be expression patterns in the muscles of Duchenne patients. It could be MRI of the heart in some of the cardiomyopathies. It could be a biomarker. And once you do that, you basically lower the bar for entry. And that's what they've done. In terms of in vivo gene editing, where this is a rising field, they've been encouraging our clinician investigators to really get into the gene in vivo gene editing using, again, surrogate type of off-target effects. So you can use human cells from different organs, for example, but all ex vivo, to basically make the point that the delivery of these gene editors are not causing harm.
Yeah.
So again, it's been a very permissive.
Could we potentially see gene therapy for type 1 diabetes, for example, which is much more, you have thousands and thousands of patients. It's neither oncology, neither rare disease, but, something very different. Do you think that in the next 5 to 10 years, we will see the application of these type of therapies for more mass type of disease? Yeah.
So for diabetes, for type 1 diabetes, there is a cell therapy solution, autologous cell therapy, either by iPS
Okay.
or the other type cells that are in phase 2 trials. So
Okay.
Vertex has a very big program. A number of other companies are doing it. So you will see it, I think, as a commercial entity within the next 3 years.
Okay, thank you. Excellent. I will actually ask Suresh the next question. Suresh, you've been working on stem cell derived exosomes as a therapeutic, not quite cell therapy, but still, I don't know how regulator is categorizing that, but what are the global regulatory requirements for using exosomes derived from stem cells as a therapeutic, both globally and in India? I mean, what are the challenges there? You were telling me during the break, there's a lot of challenges. Let's hear some of those. Yeah.
Yeah, thanks, Ajay. Just good talk. Thank you. That's a very relevant question, I would say, in the context of how people are going and getting exosomal treatments. Actually, they are going to Mexico, Honduras, probably somewhere else as well. So it's not regulated. Rest of the world, where there are no strict regulatory agencies like FDA or OECD guidelines or IHC or even CDSCO in India. But what I have learned in my own exploration of creating exosomes for certain therapies. I'm working on exosomes that could treat ocular disorders, wound healing, and so on. These are simple disorders, and there should be no contest here for actually getting some sort of approvals, because these are not systemically given. These are on surface, more or less. And because these are all immune suppressed organs in a way, the cornea, for instance, and so on, there should be lesser guidelines. But having said that, the hair transplant, the cosmetic, those fields are extensively using exosomes that are not regulated, but it's getting used from cosmetics. So there are two different branches to your question. One is unregulated exosome market is widely prevalent.
Even in India?
It is.
Okay.
It is. And most of it is done by plastic surgeons and cosmetic surgeons with imported exosomes, which are not regulated by India right now. So, okay, so that's one segment. But if you want to go for exosomes as an approved therapeutic, the guideline is very similar to cell and gene therapy or any small molecule even, and the guidelines are strictly enforced by FDA, for instance. I'm sure there are some that are approved for certain diseases or disorders, but the guideline is you have to take the molecule all the way, you have to take the exosome, in this case, the biologic, all the way through preclinical phase 1, show the efficacy, safety, and the whole 9 yards. There's
Yeah.
360 from FDA that regulates it. CIDSCO has guidelines that regulate that. We recently submitted one application to CIDSCO and got rejected, to be frank. The reason for that is pretty obvious, and which Dr. Hajar's talk also actually highlighted, is the CGMP manufacturing of exosomes. So what we are talking about is exosomes that are collected from the cord blood stem cells is where I am approaching, are very difficult to get approved because they don't fall under the autologous treatment. So if the person goes and says, I want to get treated for my condition with the exosome from my body, those are not regulated.
Yeah.
Those are allowed
Yeah.
because these are autologous and the
Right.
surgeon can do it, harvest the exosomes from the fat or any other source within the body of that person and put it back after purification. So there's no need for regulation or anything because it's done without any modification in the clinic of the hospital or the surgeon who is treating that patient on that day. And the gap between harvesting and treatment is 2 to 3 hours. So it's kind of not required to be regulated and no modifications are made.
I know
So-
when just to
Yeah.
just interrupt you, sorry about that. When you say they don't need to be regulated, that brings the opportunity for abuse, right? So
Yeah, there is an opportunity for that, but this is here we are talking about a surgeon who is boarded, certified, everything, and the patient is getting treated by him. The harvesting is done by him. The lab thing is happening under his belt. The PRP treatment comes in that category, and that's widely used, widely approved as well. And it's autologous because it's from the same person. By modification, when I said that it's not centrifuging, centrifuge is minimal. But when you put it through growth medium, there are other factors that are added. So those are not regulated and those are not allowed even. So there are, so this is how it is right now. So real exosomal therapeutic in India is difficult to get right now because they have to go through all sorts of testing.
that's good though,
Yeah.
right?
It's good,
I mean,
it's
it
good.
protects the
Yeah, I
patients,
support it, encourage it.
Yeah, yeah.
And I'm just trying to tell that for cosmetic and superficial with minimal modifications or
Yeah.
corrections, it's
Yeah.
approved. But for other things, it's heavily under the watch of CIDSCO, FDA. And
That's great.
TSC.
I mean, I think it's an important therapeutic opportunity. So it's good to know that India, from what you just said, it appears that Indian regulators are very well aware of what needs to get done and how to
They,
get
they
it
classified
approved.
it as a biologic, so
Yeah.
they're very well aware of it. And more importantly, I just want to add one point, Uday, is the CGMP manufacturing.
Yes.
That is the hard nut to crack for exosomes. And,
Yeah.
and that's my experience.
Excellent. Thank you, Suresh.
So, autologous is approved, it's the same.
My next question is to Subra. Same question, Subra. You've been at NBM, you've been at BIRAC. Where are we in terms of, approvals of cell and gene therapy? Are we way behind the United States? I know CAR-T has been approved. A couple of products have been approved. I have not heard of any gene therapy product being approved at all. So where are we in India versus, rest of the world in getting these approved?
Right. I think that's a nice question. And of course, 1 year I just came out of BIRAC, but just to mention that definitely the region or the area that we are going in future is the cell and gene therapy. That has been accepted everywhere, even all the organization, all the ministries that we have taking care of the healthcare. And that is one of the reasons that recently, like last year, we have this BioE3 policy that has come
Yeah.
out. And if you
Yes.
see in the BioE3 policy, cell and gene therapy specifically is a specific paragraph that has been mentioned. So, and I think DBT is coming up with a biomanufacturing facility, which is also providing a correct ecosystem. So, like we heard about the from Professor Hajar with respect to how they're creating the ecosystem in MGB. Similarly, we have another ecosystem which is called the Catapult in UK.
Yeah.
And I think we are following those models, and the government of India is following those models to create some biofoundry,
Yeah.
which can support the companies who are working on the cell and gene therapy. Second thing is that I think this has also been realized, even if it is complex, but it is not at a very large scale that
Yeah.
you work like
Yeah.
for biosimilars or any other therapeutics. Like I think we are way behind the China or the Korea comparatively because of the scale of operation. I think Korea, if you go the 100,000 scale, is easily available, whereas in India it is still limited to few companies. Not everybody can afford with respect to the capex investment. But cell and gene therapy, I think it is not the capex. It is the technology that you are doing. I think it is other things. What are the things that we need to make ourselves cost effective? And one of the things I think Roger mentioned in his last slides that indigenous
Local, yeah.
locally produced everything.
Yes.
We were in a conference, I think cell and gene therapy conference in CMC Vellore, I think it's last year. And there, the major cost for the cell and gene therapy is coming from the cytokines. 70% is coming from the cytokines. And I think
You mean for profiling cytokines or?
whatever, I mean, if you are doing yes, for the in the growth media or whatever you're using the
Yeah, yeah.
cytokines
The
for protein
contamination. Yeah, okay,
right,
I got
growth
it. Yeah,
factors,
yeah,
the
got it,
cytokines that you're
got it.
using.
Yes.
Plasmid DNA is one of them. There are monopolization of certain companies who are only manufacturing those. And I think if we can produce them locally, definitely we can reduce the cost significantly. And we have to reduce the cost because it is not going to be, if it is not going to be affordable, it is very difficult.
Yeah.
Today, I think you have seen the slides where, I mean, in US or in other places, with respect to cancer or the rare disease,
Yes.
the number of research is going on, number of molecules are going on, it's very high. And rare disease, honestly speaking, with the population in India, it is not rare.
Yeah.
So definitely,
It's a US definition.
it's a US definition. it's a rare-
200,000 or
exactly,
whatever,
yes.
yes.
We do have huge amount of population who are also suffering from the similar kind of disease, where I think there is no treatment available.
Yeah.
And if we target those, I am sure that government is going to support the full-fledgedly with all the companies that is coming up.
So I was pleasantly surprised how quickly we allowed companies to commercialize CAR T. I mean, it's a pretty complicated, maneuver to do that. Were you surprised at how, I mean, what is driving this speed in India? I've not seen this kind of speed before by regulators, but when it came to CAR T,
You're
it
right.
was there and there's like half a dozen of them that's going to come out. So what is the motivation there?
No, I think the motivation definitely.
I mean, is that affordability of cutting edge gold standard therapy or?
To bring India into the map of the
Okay.
CAR T, definitely we need to have. So if you now see for the vaccines, for COVID vaccines is one of the examples that we
Yeah.
came into the market very quickly. And safety, they initiated a program is the 100 days, the start from your development to the market.
Yeah.
It's a 100-day program for the vaccine commercialization. Now, these are all that getting discussed, and of course, the regulation is one of the key factors that where it takes much more time to understand, to review, and then finally come out with an approval or a non-approval, the decision.
Yeah.
So that, I mean, the government or the CDSCO is actually improving in a significant way. So they have increased the manpower, increased with respect to the review system,
Okay.
the training coming out from the different other regulations. And these are the things, I mean, it is coming in all the sectors.
Okay.
I mean, all sectors to improve or fast track the approval in
That's good to
drastic
hear.
times.
Very good to hear. Dhananjay, shouldn't you be in a class? You look so young to be sitting here. You should. Well, same question to you. In terms of cell and gene therapy, as Shubhra said, India is making rapid strides. In cell therapy, yes. In gene therapy, I have not heard anything. Would you like to comment why that is that we are not hearing so much about gene therapy in India?
So I mean, maybe I mean I do think there's some activity that's starting to happen.
I know that's exactly what I'm asking.
Right.
What is it that we are not doing?
Right.
I mean, if you can do cell therapy, I would think gene therapy should be equally possible, right? But I'm not hearing that. So I thought maybe it's a regulatory hurdle we have, but what is going on? Do you know?
Maybe I mean I've mostly been so I'm based in Boston, so I'm more aware of the ecosystem there and like
Yeah,
unlike
that we
India.
heard through Roger,
Right,
but
right.
okay. Okay.
But again, these
Well,
gene
if you wanted to speculate, what could it be that India has, that's an area we're really fallen behind. So,
right. So in I even if you I if I had to speculate, I think one of the things could also be the patent landscape, right? Because in terms of, for example, CAR-T, you are targeting, for example, the two CAR-Ts that have gotten approved are for the same target, like CD19, but they were able to go in with different sequences to target CAR-T, right? They were without infringing each other's patent.
Yeah.
But in context of gene therapy, there are two main things that go into gene therapy, right? What like Rogers spoke about, it's the capsid and the genetic payload that you're delivering,
Right.
right? And in terms of the capsids themselves are patented, so it's very to develop a new capsid altogether is a very complex process, a long process. So unless you are able to license it out from somewhere or develop your own.
Couldn't we innovate on our own? We don't have the capability to innovate.
I definitely think we do, but
Okay.
I mean it's a long process, right?
Yeah.
For example, Roger himself has developed some capsids. It
That's
takes numerous years
Right.
to and I think that process is happening. For example, in Bangalore, there are a couple of
Yeah.
labs, Arka Shubhra,
yeah.
in Delhi, there's a
Yeah.
lab. So
Yeah,
there's
yeah.
work happening, but I think maybe the gene therapy product development cycle is maybe much more complex as
Yeah,
compared to the cell.
I mean, it sounds like there's a technology gap in India
Right.
that we need to overcome.
Exactly. Right.
Yeah. Well, great. Thank you so much. I'm going to ask Noor now, who is our very well known for his work on vaccine. Noor, how are we? I mean, COVID has put India on vaccine, and I believe the numbers are 40% of vaccines globally are supplied from India and all of that. But where do you see gaps between the US and India in terms of commercializing, getting vaccines to patients, for example?
Yeah, first of all, thank you. And thanks, Roger. Wonderful talk. It's enlightening. I think next Moderna thing is going to happen. Okay. So in terms of research and in India's, in context of Indian scenario, see, we compare NIH funding.
Right.
It was almost like a $2.7 billion in 23-24.
I think it's going to be cut back now, is what
I know that's but so still in India we need
Yeah.
basic without basic science we cannot have a translation. So as academia we see that a lot of fundings is required. Okay, and I just now heard that India's SEPI program they have started that within 100 days if some pandemic comes we should have a platform to have a platform ready so that within 100 days we can have a vaccine. So for that we need funds, and for that government has initiated several programs for funds like Bio3 program. So many are there.
Yeah.
But in terms, I think if you see the rise of vaccine industry from smallpox era to COVID era, there are a lot of platforms have come. And if you see the mRNA vaccine is evolving, even Genova Bioscience, they have done a lot of
Yes.
are in
Yes.
clinical trials. And but still, in regulators in India needs to be quick because we have a bad experience during our COVID-19 that
Yeah.
our antidote. It took a lot of time from preclinical to clinical phase 1 trials, getting approvals. But I think things are improving, and yeah. So
So you think the gap in vaccine is not the technology part like in gene therapy, but more funding and regulatory hurdles?
yes, funding
Okay.
in the terms, I think industry also, they have to show the big hearts to come to the academia, give funds. Okay. So because
That's the chicken and the egg
yes,
situation.
that's true, but still because now government is also putting a lot of pressure that you have to go to industry
Yeah.
and seek the funds. So I think in that sense, the bridge has to be like academia industry bridge has to be narrowed down. And
Yeah.
hopefully, I think uh, the major that the platforms for vaccine discovery, which is either in terms of antigen selection or antigen delivery or adjuvanting, it has to come from the basic science.
Okay.
So
Understood.
industry has to show the big heart.
Yeah.
They have to come,
Yeah.
I think, yeah.
Yeah.
But there is a remarkable, if you see post-COVID, a lot of platforms
Yeah.
are
I
coming.
know. It's
In
amazing.
terms
how
of
well.
adjuvants, okay, we
Yes.
ourselves have made some of the adjuvants. We're able to translate, the technology has been translated to some of the companies.
Yeah.
and these
Yep.
are in the pipeline.
Excellent. I mean, like you said, it's not technology, but there are other
Yes.
elements
So,
that we need to
Yeah,
get.
regulators like
speed.
CDCO, DG said, and now the
Yeah.
timelines are coming down. And slowly improving. I think.
Okay.
Yeah.
That's good. So I will not ask 3 questions. I'll only ask 2. I'll start with Roger again. Roger, what are the safety concerns that a patient should have in all of these technologies? I mean, are we doing enough to make sure that by the time, for example, for cancer, you might want to get something out which is even half baked because the patient has no other option, right? And that's probably true for other therapies too. So if I had to ask you, what is your concern about safety? What's the number 1 on top of your list?
Immune response, toxicity from the immune reaction to in the gene therapy space, that's really our big concern. And that's also technology related because the amount of vectors that we deliver are astronomical.
Yeah.
That's where the cost comes in, and that's where the safety. We have a whole medicine category of AV related toxicity.
Yeah.
And they go from very mild to life threatening. But as you mentioned, in many of the cases where we deliver high doses of AV, the alternative is death for the patient.
Yes.
So do you take that risk of delivering very high doses, giving immune suppression at very high levels while trying to cure the patients? So there's always that balance. It's very
Yeah.
delicate.
Yeah.
What's going to solve it is innovation in technology. We need better vectors. They're going to be more effective. So we have to give less of them and elicit less immune responses. Vectors that can evade the immune system, ones that detarget certain organs so that they just home in on where they're supposed to go. Because a lot of the immune response is because you're getting these vectors in the spleen and the liver.
Right.
So again, there are multiple wish lists
Correct.
technologically that we have. We need to execute
Yeah. Okay.
to get the safety levels.
Excellent. Yeah, thank you. So same question for you for vis-a-vis exosomes. What is your nightmare safety risk?
Well, the characterization of the exosomes is a technological challenge. Because of that, the risk is the exosomes, when they are harvested, they can come from good, healthy cells that secrete very good exosomes with perfectly fine mRNAs, DNA, cytokines, growth factors. But there could be other senescent cells or zombie cells or dead cells that also could have
In the
exosomes in the picture, in the preparation. So the biggest safety risk is the purity may be lacking, and that could lead to no effect or drastic severe reactions or effects. That is the challenge in exosomes right now.
Okay. And the way to standardize that is to have a checklist of saying you need to do this, you need to show this, and okay.
Exactly. So when I mentioned GMP, that is exactly what was missing in the portfolio that we submitted. Is they wanted us, for instance, there are several ways to characterize. We go with size characterization or molecular characterization or FACS.
Yes.
But they would ask for much more stricter, specific characterization, not just based on size, maybe their binding abilities, antibody characterization, and so on.
Okay.
So that technology and innovation is needed to perfectly characterize them.
Excellent. Thank you. Shubhra, what about cell therapy? What is the nightmare scenario you imagine as a risk for cell therapy?
I think for CAR-T already there are certain elements that has come up for the long-term effect with respect to the I think the autoimmune T cells or something.
Right.
And there is a
T cell exhaustion.
Right. And there's now FDA has introduced a black box warning into the any CAR-T product. So again, these are the newer technology, and definitely with respect to the safety evaluation, I think there is no shortcut that to be applied. So that's one important thing. I mean, today, if we're, as we see that we are moving into a different regime where, we are not using animals, of course, we're going into organoids or cell-based testing for the preclinical.
Yeah.
So you are avoiding one step. So definitely your clinical testing has to be very robust.
Yeah.
Need to have more biomarkers that are available which can evaluate the clinical results more efficiently. So that's the area. I think in this probably the AI today we are talking about the integration of the AI, and I think here I love to see that a lot of companies are working on that platform. And I think for this, for the clinical trials also, maybe integration of AI is important, which can predict much more what we cannot just see by evaluating simple statistical analysis.
Yeah.
So that's what my thought process is.
Okay. I mean, do you ever see cell therapy being adopted for more chronic diseases? I mean, again, cancer is, as we've discussed, it's a no hope situation. So yeah, go ahead and do it. But is it safe enough for cell therapy to be one day used for chronic therapies, chronic diseases?
I think one of the major factor I feel, I mean, cell therapy or the gene therapy, maybe if it is the final or the ultimate therapy, yes, it can be. Unless we reduce the cost to a significant level, I think even if we develop anything, it is not going to be, we cannot commercialize.
Cost is not a safety risk. So yeah, but I understand what you're saying.
Right.
Yeah. Yeah. Thank you. How about you? Gene therapy, what is your nightmare scenario? Oh my God, I should not have done that with gene therapy.
Yeah, I mean, I think some of those are already mentioned, maybe, but maybe I'll mention a couple of those, maybe not on top of my list, but those that have not been mentioned. One is maybe some of these vectors, for example, integrate into your genome. So having the right QC to make sure that there's no unintentional mutagenesis, right? I mean, for example, the genome that you're trying to integrate the payload, maybe, for example, near some oncogene leading to oncogenesis or something. So that's one. Second is, I mean, a lot of these cell therapies are immunosuppressive in nature, and so that may be secondary infections in patients. So monitoring patients for all. Maybe these are a couple that have not been mentioned.
Can these be teased out before you go into a patient, or you can only understand these in a human situation?
I think the first one definitely, I mean, using advanced sequencing technologies now, there can be QCs, especially for the ex vivo therapies, right? Maybe if the therapies are in vivo, it could be a little bit challenging to see where in the genome has the payload integrated, but at the same time, for in vivo, generally we use vectors, for example, AAV, which don't integrate.
Yeah.
And for the second one, in terms of infection, I mean, what was earlier mentioned, using GMP to make sure there's no contamination leading to infections in patients, having high QC, that can be integrated.
I have to tell you, there's at least a half a dozen companies in India that are doing gene therapy now.
Right.
They're below radar right now, but there are companies that are doing it.
Right.
Right here in Hyderabad, there are a couple that are doing gene therapy. So I think it's just a matter of time before you start hearing.
Definitely.
Yeah, 100%.
Yeah.
It's happening. Yeah. There's one gentleman who just walked out. I mean, his own company is doing that. So, yeah. So, please, now what is the biggest risks in vaccines? There's so many anti-vaxxers, as you know. Autism is a risk, blah, blah, blah is a risk.
So many,
Where do you stand on vaccine being a risk?
so many negativity out of vaccines. So we can see in the newspapers also. So
it's become a political issue, right? The anti-vaxxers and all that. Yeah. So is that all real, or people are imagining, or where do you stand on this?
So we have scientific data which shows that it protects 150 million so far.
What vaccine is this?
No, total from the smallpox until now, the data 150 million lives it has saved. So it has a protection.
Yeah, this thing was with the human papilloma virus. There's a lot of brouhaha.
HPV, they are saying, but see, there are a lot of side effects, and it could be one of them is the what we are talking about, unwarranted effects, what we are observing by the vaccines. Okay, so maybe the backbone what we are choosing, inflammation.
In the adjuvant, or
no, for example, if you see untargeted, like when you give a nucleic acid, mRNA or DNA, untargeted effects. So that's how the platform is required to have a, for example, you target dendritic cells, immune cells, and that's how you suppress the inflammation.
Okay.
So that is one of the big side effects, which in terms of heart attacks and all those. But now there are a lot of modifications as circular DNA has come, which may, or there are technologies are coming where only you target dendritic cells. You
Yes.
engineer such a way that vaccine only go to immune cells, not non-immune cells. So
Okay.
that way you can curtail inflammation.
Kind of you target using antibody.
Engineer like exosomes, what we
Ah,
have been discussing, you
okay.
make an engineer exosomes target to dendritic cells. So these platforms, many companies are coming, vaccine companies. And yeah, so, safety is always a concern with the vaccine.
Yes.
And with so many, even the GBS and so many new things are coming, and they're correlating, okay, because of molecular mimicry, you're putting so much antigen and can have cross reactivity, and that is leading to disease.
Have we found any link between the COVID virus and there's so many long COVID effects? Do you think those are vaccine related or just the biology of the precursor?
See, that's the thing. See, there is so much molecular mimicry between virus and self antigens.
Okay.
Anything can happen. So that's why it's moving to a subunit vaccine. So you don't need a complete set of entire virus. You just take a one component, immunogenic component, and either in the form of mRNA or protein, and use that. And many US anti-vaccine group is working.
Yes.
So I don't know how to discuss about that.
Right.
So,
But I mean, from what you're saying, better understanding of the molecular biology of the vaccine itself and what you're targeting, you think is one way to make sure that you don't see any significant side effects.
yes. So what I suggest is to minimize the antigenic load.
Okay.
So not to give a lot of antigen, just give
Okay.
the antigen which is required. So either you have a sustained release,
Right.
and with good awareness of immune responses. For example, adjuvant engineering is coming very good. Okay, so good adjuvants are coming, which cut down the requirement of antigens.
Yeah.
And that's why it's suppressing most of the live attenuated vaccines.
Yeah.
And ad vaccines are also problematic, viral vectors. With what we have, Roger has been talking is ad vaccines, you have a lot of antibodies generated against the viral vectors.
Yes.
So that is again a problem. So
Excellent.
I think
Thank you. I think what we should do now is the floor for some questions. And if there's time left at the end, we can have them each say, what they think is at the top of their list in terms of what's important. Yeah, please go ahead. Is there a mic?
I have one question.
Go ahead and ask it, please.
Yeah.
Let me also know who you want it to be answered by. So.
Maybe I'll just ask in general. So,
Okay, sure.
so what is the utility of a cell therapy in the neurological disorders? Because I feel like, unlike other disorders, just refilling the number is not good enough, right?
You mean for stroke, things like
For
that.
Alzheimer's, or
Okay.
Parkinson, where you lose the neurons,
Right.
and just refilling the neurons doesn't help much because the memory and all these things stored in the form of connections rather, right?
Yeah. It's a good question. Yeah.
Yeah, I
Roger.
mean, no, I think definitely an important question. So neurodegenerate, I mean, if you take neurodegeneration, it's a pretty complex pathway that's been targeted by multiple antibodies, gene therapy. The simpler model is the Parkinson's disease, where you're losing neuronal cells in a specific area. And that's where delivery of iPS-derived neuronal cells that can generate the specific, GDNF, for example, in the brain is able to reverse some of the Parkinson symptoms. You saw with the very specific gene therapy for AADC that you're able to reverse the Parkinson's or the precocious Parkinson's in these children. So in neurological diseases, there are some very specific examples where cell therapy especially in Parkinson's, can be addressed. And there's one company called Blue Rock that's using allogeneic neuronal cells to inject in Parkinson's patient, and they just entered a registrational trial. So this is their phase 3 trial, and they're very bullish about it.
Yeah, thank you so much. Yeah, please go ahead. Yeah.
Thank you. Very nice presentation. My name is Parag Kinge. I have worked in Genova, and that was a mRNA project which I was part of. So,
Can you hold the mic closer to you?
yeah, the basic question I have is about animal models for gene therapy. I mean, since most of the genes are human genes, which we are targeting, I mean, are there animal models, enough animal models where we can really test those things instead of going to clinical trial because it is much cheaper there? And second question is, there are a lot of intermediate things required. For a final product, you definitely have, but you have reagents, for example, enzymes and those things. In case of mRNA, when we were developing that product, the cost of the enzymes was very low at the beginning. And all of a sudden it went, I mean, almost 20, 30 times. So the thing is that was a hurdle. So we can't really sell that product when other products were available for 50 rupees, 100 rupees. We were not able to sell it. So are your institute also providing services there where how to vertically integrate things and help developing products?
I'll take the first part of the animal model question because I'm a veterinarian.
Yeah, sure.
To begin with. So there are such animal models. In fact, there are many viral oncolytic viruses that were using animal like vesicular stomatitis virus, and very, very similar to human viruses. And they have been programmed to act as tumor killing viruses in the humans as well. So they can kill viruses, can kill tumors in birds and animals as well as in the humans, the same virus. So there are several animal models for understanding gene and cell therapy. Yeah,
Great. Yeah. So in terms of the cost of these enzymes and other plasmids that are necessary for RNA production, so our institute tries to, as an academic center, we get big discounts. So we're all, since it's not commercial, we always have that lower cost. But I think from an Indian market perspective, it is so critical to make the raw material, whether it's enzymes, the plasmids, all that locally, so that you're able to benefit from low costs. Otherwise, as you said, these companies can charge whatever they want, especially if they hold monopolies, and many of them do, unfortunately.
Yeah, we had just there was
Just hold the mic closer. Yeah.
there was discussion about gene therapy. The CMC Vellore, there is a Professor Siva who is developing this thing. He had come to us, and then maybe quoted him some things about developing the plasmids and all those things. He said, "I cannot, I will not able to even treat 10% with that code." So that was really a kind of a problem, especially in case of hemophilia. In India, it is a huge problem. So they are developing some technology there.
Yeah.
Yeah, so maybe I'll take this opportunity to talk a little bit about the efforts that we are doing in Bangalore at VJ Bio, because we're actually trying to solve that problem. So at VJ Bio, which is a new company, I mean, new as in it's a startup company, we have been working in a stealth mode for past 2 years and getting ready to come out of stealth mode. And the problem that we are trying to solve is how we ourselves are not interested in developing therapeutic products, but how can we enable the innovators who are working on developing cell and gene therapy products? How can we support them with these critical raw materials, various grades of critical raw materials, starting from R&D to high quality to GMP grade, starting from plasmids to viral vectors to enzymes. And so that's what we have been working on for the past 2 years, and we're ready to commercialize some of these products starting next month. So happy to talk about it.
Yeah, thank you. Great.
Yeah. Go ahead, please.
Sir, my name is Suhas. I'm from Osmania University, and my question is like general question to everyone. Anyone can answer. Sir, now normally what we are seeing is personalized medicine because more and more we discover that we are seeing more and more specificity to the target, because many people suffer. We are seeing genome variants and everything. So in terms of personalized medicine and coming to CAR cell therapy, especially now what we see is like we know the side effects of the first gen CAR. So we are developing second generation and third generation, like providing, as you know, like we are trying to provide logic gates also for spatial temporal activity, so that we don't have off-target toxicity, especially. So I have a question that if you are increasing the specificity of a disease in order to prevent off-target toxicity, and at the same time trying to cut down the cost, how is that possible? I mean, in terms of innovation, as you speak, we need vectors, payload less, and everything. So is any like speculation of how to reduce? I know it's an innovative model, but any chances of like reducing, cutting down the cost and increasing the more specificity? So how is that possible? How to break down that paradox? Yeah, thank you.
I can give my 5 cents on that one.
Okay.
One is I did work on PD-1, PD-L1, and CTLA-4, the checkpoint inhibitors. So one of the recent trends is companion diagnostics, that is measuring the biomarkers, specific biomarkers that can get elevated or reduced as you provide the treatment. That makes personalization a little bit more easy. And there's pharmacogenomics that is kind of exploding in the field right now, where you can actually pick the right patient for the right kind of therapy, so you'll not have the side effects. And the third mode of activity is, I want to use the term docking, which you actually briefly, I mean, India just successfully docked the space satellite, right, upstairs. And we have Jibin sitting here, who is planning to send some biological payloads into the space. So some of this so we are advancing very fast in all these innovative areas that can cut down the toxic and strange reactions that can happen. So this is the extreme innovation that is happening right now by way of docking, homing of cells, for instance. Cells would like to go to a particular location because they have the receptors there. But the dendritic cells and macrophages, they can take them somewhere else.
Yeah.
So these are challenges that we have, and there are innovations happening in this. And this is specific challenges associated with cell therapy, not gene therapy, but stem cell therapy and exosomal therapy.
Yeah.
But there are innovative ways of treating this kind of challenges that we are facing.
Great. Roger, did you also want to comment?
Yeah, I mean, so yes, it is a little bit of a paradox. Innovation costs a lot of money. And how can we say that by making it more specific, you're going to make it more cheaper? I think the answer is that if you make it more specific, the amount that you need is going to be much lower. So the cost of goods is much lower, and that offsets the cost of the innovation.
Yeah, excellent. Anybody else wants to, yeah, no? Okay, any other questions we have? Yeah, please go ahead.
Hi, my name is Surit. I'm from Avaya Biosciences. My question is around the valley of death that you talked about. So I wanted some of the speakers to share their insights on that. How do innovators what are the kind of checklists, an innovator needs to have to get past, animal studies and phase 1, phase 2 trials? What are the things that you've seen in your experience that's pretty common for most therapies in terms of either, certifications, paperwork, or financials? Like, what is the quantum of money that you'd need to move?
So, so this is one question I've spent all my life thinking. I'll tell you one thing. There are certain diseases where the risk is completely disproportionately high. CNS is one. Animal models don't translate very well. Unfortunately, oncology is the same way. So one way we have been de-risking, or the industry typically de-risks, is to go towards areas, metabolic diseases. Translation is fantastic. Really high in animal model, and what happens in humans. So one way to de-risk this value of death, as you said, is to be careful about choosing which indication. Don't just go after, oh, it's so exotic that I'm going to solve brain tumor, right? I mean, the industry doesn't work like that. They work more towards, what's feasible, what's risky, what's not risky, and all of that. So I think some thinking upfront about translation of what happens in animal models into humans, you have to severely think about that. Not just get too excited about the idea, but Why do you think all the large pharmas have something called herd mentality, right? If you watch the starting class of drugs that we have 8 companies have produced the same, that's the herd mentality because it's very obvious for Pfizer, so Merck will do it. Somebody else will do it itself that you need to be really careful. Yeah. Anybody else? Yeah.
There's also, I mean, I totally agree with Uday on that one. There are newer models that you can use way ahead of going and thinking about phase 1 or preclinical in the animal model using the appropriate cell lines. And actually, don't get too attached to the molecule when you discover a molecule, think that, okay, this will cure everything. Understand the structure activity relationship of the molecule from the literature extensively. Use a lot of AI and algorithms to understand how it, and Uday has done some work on this as well. So use those things extensively so that your risk of losing the molecule or spending too much money on that molecule is de-risked.
Yeah.
Yeah.
Well, one other thing, excellent points, I was going to add is go after targets that are validated. When I say validated, if there's a gene polymorphism in humans for that target that reflects in a, if the genotype reflects in a phenotype, then that's a validation from disease. So validated targets offer you a much lower risk rather than unvalidated targets. So there's like a laundry list of things that large pharma has before we go after anything. We're not just driven by exciting science. That's probably at the bottom of the list, to be honest. But there's other de-risking things. We should have a conversation offline on that. Yeah, sure.
Maybe I just want to
Brian,
add,
please.
yeah, like Sanofi for the dengue vaccine developed, and it failed miserably in the clinical trial because they have not, developed one assay, right, which is going to select the right patient. So that is what, okay, 30 years, it's a very long time. I think we don't spend that much of time. But remember one thing, that don't rush to the clinic because you need to do lot of development, lot of validation much before, because do as much as you can to de-risk your molecule before you go to the clinic. Because honestly, in human, like, again, I'm giving an example, the CMC Vellore conference, MHC, MSC, mesenchymal stem cell, there are so many molecules are coming out, so many clinical trials, phase 1, phase 2, fantastic result. Phase 3, failing. So once it goes to the human with a bigger population, it's a totally different story. But we need to see what are the tools that we have in our hand to understand that better, and then you go to the clinic.
Yeah. Yeah, we'll take one more question. Then I'd really like to have Roger's views on, you've heard a lot about where Indian science is in terms of gene, cell, and so just your 100,000-foot view of where we are and where we should be going. So we'll have take a last question, please. Yeah.
Hello, sir. So one of the faculty coming, sir, he said that simply filling the gaps in the brain, like in case of the substantia nigra Parkinson's, won't be enough. And so I said it's positive results. It's in very last stage of it's very responsive, right? So there can be like a combination therapy, like including the cell therapy and the neuroendocrine modulators can be used to guide, or in other cases, in other diseases, can it be a combination of therapies, including cell and gene therapy? I want your comments, sir. Thank you.
Maybe Roger can.
So there are antibody drug conjugates, combination therapies available, but maybe Roger can explain.
Yeah, I mean, cell therapy, CAR T therapy is a combination of cell and gene therapy. So we're genetically modified cells to deliver one of the most effective oncology treatment, which is CAR T. So that combination is already has been working. You'll see more modified cells coming down the line because you're going to make them more immunosilent. You're going to need them to be more effective, targeting specific areas. So that combination is going to continue. Whether it's going to be like we're doing it now, ex vivo, you deliver the cargo and then deliver the cells, or it's going to be cell delivery with a combination of RNA therapies. It depends on the disease state, but that's definitely coming down the line.
Okay. I think we'll just have last a couple of minutes of your views. And you've heard and seen a lot of cell, gene, and vaccine experts here have told you where we are in India. What's your 100,000-foot view of where we are, and are we heading in the right direction? I mean, for lack of better words, yeah.
Yeah. Is a very exciting field. I wouldn't be in India so many times. This is my 5th or 6th time in the last couple of years if I didn't think India had a huge potential in delivering on the promise of gene and cell therapies. There are many hurdles, just like with anything else. But I view the ecosystem here with young investigators, with the really appetite to get into the field as some of the key ingredients to making this a success. In the West, especially in the US, have really been the costs of doing, the various steps of going through the various steps in gene therapy and cell therapy, and also the regulatory framework. I think that lowering the especially raw material is going to be very important. Innovation, I know sometimes it's costly, but at the end of the day, innovation pays off. And the more you innovate, the better product that you will end up with. And finally, I think that there has to be the political will to change the regulatory responsiveness in India. Clearly, it was done for CAR T. It has to be done for other types of disease states where they accept that gene and cell therapy is going to have surrogate endpoints, and the pathways have to be, open for that. So there are still obstacles, but I view them really crumbling down as the train's going forward. And the train is going forward. There's absolutely no stopping it. And I think that India will be a leader in this field in the next 10 years.
Excellent. So when are you going to set up a company here?
It's already there.
That one. Okay. No, I meant in Hyderabad. These guys are not in Hyderabad. Yeah, Nalini sir, when you stepped out, we were talking about gene therapy companies in India, and I said there's a lot of activity that's going on here too. I know that's close to your heart, so would you like to comment on? Should we get you a mic? And because really like to hear people in India who are doing gene therapy type of things, because I think it's pretty pioneering. So.
I had a good discussion with Roger and his team, and we were talking about our major bottlenecks of this reagent supply chain and all that thing. It's so expensive. Even to do an R&D trial, it costs you several lakhs for each experiment.
Right.
That's why you see slow in this process. So we are actively pursuing a couple of products in the last 2 years. So we were discussing about how to make things fast track. So one of the things is basically availability of these reagents. Now there's a monopoly of only 2 or 3 companies, very expensive, whether it's media components, cytokines, those magnetic nanobeads coated with antibodies. So I think now there's a lot of activity going on. There are a lot of startups all over. I think we see a significant progress in next few years. You will see more and more products coming.
Is there risk appetite, sir, in our company innovators here?
That's why we take up only those generic products, the similar products that is already approved in US. That's the approach we take. There are now more than 10, 15 CAR T therapy products already available. So that's what these companies in India, including us, looking at it. So the only thing is the cost of development, cost of clinical trial is still very high. So we are working out the backward integration ourselves and few other companies. And VJ Biotech is also, it looks like, in the same area.
Excellent. Thank you so much. I think it's 12 o'clock, and a round of applause for Roger and everybody here. I think all of us learned a lot about some very exciting, areas that will be around for the next, I don't know, 50 years or so. Thank you so much, guys. Thank you so much.
Thank you. Please stay here. We have mementos.
Oh, okay, okay.
Dinner, sir, please come.
Yeah.
Sorry.
Gaurav?
Oh. That's-
Camera on that. Yeah, good.
Thank you. Thank you very much.
Yeah, I have to give that to Uday Saxena. Yeah.
Then you have such good.
Dr. Suresh Pusala. Yeah. Dr. Suresh, sir. Yeah. Nuruddin Khan. Dhananjay. Ashubra Chakravarti. Thank you. Thank you, everybody. Yeah. So, proof? Yeah, sure. Ranjan? Yeah. Murli sir. Sharma sir. Then you like to put it on LinkedIn. Thank you, everyone. The stack outside. This conference.